Hey everyone, welcome to lecture 16 on immunity and the lymphatic system. Now, the immune system, which is where we're going to start, can be kind of a misleading name. And the reason is the immune system isn't an interconnected system of specific organs and tissues. Rather, it's a distributed system of cells and some particular tissues and organs scattered throughout the body that are all serving the same function of defending you against external invaders. Now, we've already talked a little bit about immunity in the context of blood cells. Remember, leukocytes are our white blood cells. They're responsible for defending our blood supply against foreign pathogens. We talked about how there are granular and agranular leukocytes. What we're going to talk about now are the specific functions of those leukocytes, starting with the granulocytes, the granular leukocytes. Remember, there are 3 examples of granular leukocytes. The first of these 3 are the neutrophils. Neutrophils are the most abundant of your granular leukocytes. You find them in tissues, you find them in your bloodstream. They're highly, highly mobile. They really make the rounds. And their primary function is to fight bacterial infections. Now, neutrophils tend to be a one and done kind of defense. A single neutrophil, once it encounters some sort of invading virus or particle, will engulf that bacteria, sorry, not virus, bacteria. It'll destroy it, and then the neutrophils have a tendency to blow apart, okay, into tiny little pieces. So they self-destruct after they've encountered a bacteria and destroyed it. You have likely encountered the sad remains of neutrophils because that's what pus is. It's these dead remains of neutrophils that have been fighting a bacterial infection on your behalf. Next up, we have the eosinophils. These guys tend to be immobile. They're stationary. Oftentimes you find them in the walls of your gastrointestinal tract. That's what GI stands for. This is your digestive tract. Okay? They have a number of different jobs. Their primary one is to fight parasitic infections, so to fight any kind of foodborne parasites that you might be exposed to. They also have the ability to release a chemical to reduce inflammation once an invading parasite has been subdued. And then finally, your basophils. Basophils are awesome. These guys do not perform phagocytosis, all right, so they're not going to engulf an invading cell and destroy it. Instead, they release chemicals that promote inflammation and are primarily responsible for what we think of as an allergic response. So the functions of inflammation are maybe what you think, maybe not. We're going to discuss them in a couple slides. Inflammation is actually a way for the body to recruit more white blood cells to the location of a potential infection. It's kind of cool. So again, remember, you need to be able to identify what these guys look like from images of slides. Remember too that the giveaway telling you that these are granular leukocytes is here in the cytoplasm, right? It's all of these tiny little dots, these little crystal looking grains. All right, next up, let's talk about our agranulocytes, our agranular or without granules leukocytes. There's only 2, the monocytes and the lymphocytes. But remember, these are our phagocytic white blood cells. These guys go around and engulf invading pathogens, digest them, and destroy them. Okay? Monocytes are our largest white blood cell, and most importantly for this lecture, monocytes are what mature into a cell we call a macrophage. Macrophages are a general hunter-killer cell. So macrophages, they don't hunt specific bacteria or specific viruses, but anything they encounter that is non-self, that they recognize as not being a part of the body, they'll engulf and destroy. Lymphocytes are another type of immune cell, and they are going to become very important when we start talking about adaptive immunity later in the lecture. The lymphocytes often are found throughout the lymphatic system. That's part of where their name comes from. You mostly tend to find them affiliated with lymph tissue, but they are fairly mobile and they will make the rounds. Remember again, giveaway here for identifying these, you don't have any of those grains in the cytoplasm, right? Also, remember your nucleus is nice and round and continuous in the agranular leukocytes. All right. We're going to go over some background information now, and we're going to use our knowledge of those leukocytes in the context of the different types of immunity that we express as humans. Okay. So first, let's define some terms. The word immunity has a number of different definitions, but the simplest one, and the one we'll use, is that immunity is the capacity or the ability of the body to combat diseases and fight infection. And these are diseases that could be caused by bacteria or viruses or parasites. Toxins can even fall into this category. Your immune system, again, isn't a specific system of organs. Instead, it's a collection of molecules, so things like proteins and cellular signaling chemicals, unique individual cells, specific tissues, and organs, which combined provide protection against microorganisms, microbial toxins, there's the toxins, and interestingly, even tumor cells. If your own cells are behaving in a way counter to your own survival, your immune system has the ability to target your own body's cells and destroy them if they are behaving in a way that causes disease. There's two major types of immunity. It's what we call innate immunity. Innate means you're born with it. It's present at birth. We tend to call innate immunity systems the first line of defense. Okay? It's in quotes because it's sort of a hokey way to, well, what the first line of defense means is this is your general system of defense against everyday threats. And I'll show you what that means. This is the first defense your body puts up against invading pathogens. The other kind of immunity is called either acquired or adaptive immunity. You might hear both terms. Just like the name implies, this is immunity that you acquire over the course of your life as you're exposed to pathogens and your body learns to identify them and how to fight them. Okay? It also tends to develop especially quickly in your youth when your lymphatic system is developing. Now we're going to talk about both innate and acquired immunity. Let's start with innate first. Think of innate immunity as being made up of a whole lot of general protective measures. These are blanket measures that protect you against a huge range of pathogens and potential invaders. These are non-specific measures, and that's important. This is a non-specific system. Innate immune responses don't target particular viruses or particular bacteria. For example, classic first line of defense would be the mechanical barriers your body puts up against the environment. Think of the keratinized outer layer of your skin. Okay? Think of, even deeper, think of cell membranes, preventing microorganisms from entering into the cell. Think of the mucous membranes that line the inside of your mouth, the inside of your GI tract, the inside of your nose, that produce excess mucus and other materials to help keep pathogens out of your inner body. Okay? Chemical defense is also a part of innate immunity. Low pH in your stomach, in part, helps to destroy any pathogens you may consume with your food. You also produce defensive enzymes. Remember, enzymes are the hardest working protein in the body. They're the ones that do most of the work. These defensive enzymes are found in tears, saliva, and other body secretions that are secreted onto, again, the surface of your body. Interestingly, your symbiotic microbes, so the bacteria and other microorganisms that live symbiotically with you on your skin and in your digestive tract, they actually occupy spaces in your body where invading microbes could gain a foothold if that symbiotic microbe wasn't already there. So your symbiotic microorganisms that already live on your skin and live in your body will fight to maintain their territory, and they will repel any invading microbe that doesn't belong and that might supplant them. So they actually work in your favor as well. And finally, inflammation and fever. These two responses are probably the most diagnostic, and I choose that word deliberately, of the innate immune system's response to an infection. These are two of the ways that we can externally diagnose that there is an infection present in some part of the body. Inflammation, we also tend to refer to just as swelling. So it's when a localized area of tissue takes on excess blood and also just body fluids, what we call lymph, and grows larger in size. Fever is when your body temperature goes up. Now, fevers, we generally overtreat. There's a tendency to start to treat a fever as soon as it approaches 100 degrees Fahrenheit. That tends to be an overreaction. Your body is generating that fever for a reason. Some of your white blood cells actually work better at higher temperature. They move more quickly, they have an easier time traversing your circulatory system and getting to the site of an infection in order to fight it more effectively. So allowing a moderate fever to proceed somewhere between 100, 101, maybe even 102, that actually is promoting your body's own natural defense against the infection that you're fighting. There's a couple other effects that a fever has. One is it actually reduces the growth rate of certain bacteria and viruses. Bacteria and viruses evolve just like many other living things, and many of them have evolved to grow at a maximum rate when they're exposed to a human body temperature. So once these bacteria make it into your system, your body temperature is their ideal growth temperature, and they grow at a maximum rate. But if you raise that temperature up above 98.6, 98.7, take it up to around 100, 100.5, suddenly that bacteria is no longer in its ideal environment, and its growth rate is going to drop. It won't be able to grow and reproduce as quickly. That's in your favor. Also, this is interesting, a higher body temperature caused by a fever, it actually causes your liver to withhold iron. So your body starts to sequester iron from your blood. It holds it in the liver. It doesn't release it for circulation. Why does this matter? It's because iron is very useful for many pathogens. It's a material that a pathogen will use from your own body and use it against you to help promote its own growth. So by withholding iron, we deny access to it to a pathogen. Okay? So it's just another line of defense, and again, that is triggered by fever. All right, so what causes all these responses? Well, you're going to have cellular involvement. The leukocytes that are involved tend to be phagocytic, meaning they're going to perform, they're going to consume, engulf and consume these microbes, these pathogens, or they are going to promote inflammation, right? Like the basophils, right? So there they are. Your basophils are going to promote the inflammatory response. Macrophages are super eaters, and neutrophils, which are, they're good eaters too, but remember, neutrophils tend to die after they've consumed a pathogen. Macrophages can keep going. Macrophages just keep eating and eating and eating. Okay? Now, when a macrophage encounters a non-self cell, so say it encounters an invading bacterial cell at a site of a cut or some sort of physical injury, the macrophage will release chemical signals called cytokines. Okay? The release of cytokines is triggered by phagocytosis of a microbe. So once the macrophage has engulfed a microbe, phagocytosis has begun. That triggers the release of these cytokines. And cytokines have a wide range of functions, but the one we care about here is that they sort of send out a distress call and summon more macrophages to the site of the infection. Okay, so boosting the immune response to that particular location. Cytokines are important. This is a very critical response in order to gather more leukocytes to locations where they're needed. The trick is sometimes you can release too many cytokines and trigger too extreme of an immune response. We call this a cytokine storm. And this is very, very prevalent in the news right now because cytokine storms seem to be common in coronavirus patients. Okay. And this seems to be what causes the really extreme reactions that are boosting the mortality rate of this virus, causing people to die. Cytokine storms are when huge amounts of cytokines are released all at once. It triggers an over-recruitment, too many white blood cells flooding an area, and all of them promoting inflammation. It's another thing that cytokines do, is they trigger leukocytes to promote localized inflammation. Now, the reason inflammation is something that's actually helpful is because when you have a normal blood vessel, you've got white blood cells floating around inside, and they're trapped. They can't get out. Okay? Normally, that's all right. But when you're fighting an infection, you want these white blood cells to be able to leave this blood vessel and enter the surrounding tissue where the infection may be hosted. In order to get them out of the blood vessel, you need that blood vessel to expand and, see if I can do this. Oh, never mind. Thought I might be able to erase small pieces. Here, instead, you want this blood vessel to expand so that little pores, little holes open up in the sides of it. And these holes are just big enough for white blood cells to leave through. Okay? The trick is fluid also leaks out through these holes. And that fluid floods the surrounding tissue, causing it to expand in size, which is what we associate with inflammation. Right? So inflammation, you get increased blood flow to an area, swelling of your vessels, which creates gaps that the white blood cells can exit through so they can enter into the surrounding tissue and do battle with whatever your pathogens are. Okay, so there's a pathogen. Okay. If you have hyperinflammation, you've got too much fluid leaving your blood vessels. It's causing too much swelling, and it's flooding tissues that really shouldn't be flooded, like the ones in your lungs. All right. Let me give you an example of what inflammation looks like when it's working appropriately. Okay? So remember, inflammation is part of your innate immune system, innate immunity. One of the most common chemical signals released by your innate immune system is called histamine. Okay? Now, let's imagine that you, I don't know, fall down the stairs. Don't do that, it hurts. You hit your knee on one of the steps and you cut yourself, right? You've got this nice big gaping hole in one of your first lines of defense, your skin, okay? And of course, because there are pathogens literally everywhere, you get some bacteria, say, that makes it in through that cut and into your inner tissue, right? So this is bad. You want to make sure that you get rid of those pathogens, capture them and destroy them before they have a chance to enter into your bloodstream, where they could then be distributed to other parts of your body. Okay? All right. Cells that are near the site of the injury can detect that the damage has been done, and these cells can actually release a chemical called histamine. And histamine, as it flows into the bloodstream, will initiate an inflammatory response. It will initiate this dilation of your vessels, of your capillaries, which will flood the area with extra blood and fluid, okay, causing it to turn red from all that additional circulation. It'll also become warm or hot to the touch. Swelling, of course, will happen because of all of this excess fluid that's suddenly in the tissues. And frequently, this puts pressure on nerve endings, which causes pain. Okay. However, this is also a good thing because it creates space for phagocytes to leave the bloodstream, enter into this inflamed tissue, and start consuming your pathogens before they can even have a chance to cause an infection. Okay. So histamine is a pretty common chemical signal inducing inflammation. All right, we're going to spend the majority of our time from this point forward talking about acquired immunity. It's a little bit more complicated. All right, acquired or adaptive immunity is very, very specific. That's the first thing you have to know. Your acquired immune system is all about learning the identities of individual pathogens and creating memories of them so your body can react more quickly if it's ever exposed to them again. Specifically, what your acquired immune system responds to are those antigens that we discussed before. Remember, antigens are chemical signals that tell the body this is a non-self cell, meaning it does not belong to you. This came from somewhere outside. It doesn't have to be a cell. It can also be a virus. It can be a parasite. But these antigens are a chemical signal to the body that this thing isn't a part of you. Okay? Specifically, there are locations on an antigen. So let's just pretend that this is an antigen. There are specific locations called epitopes that attract antibodies. Epitopes and antibodies tend to have very, very tight relationships. So a single antibody will tend to only bind to a very specific epitope. They're not necessarily one-to-one relationships, but they can be. Okay? These epitopes actually attract their specific antibodies if an antigen with that epitope enters into the body. Okay? Antibodies serve a wide variety of functions. In fact, let's skip ahead one slide and talk about them. Actually, we'll talk about them in a minute. On the next slide, we'll discuss what those functions are. First, though, let's finish our overview. Let's talk about the two types of acquired immunity. There's actually two subgroups of your acquired immune system. The first is called humoral. Second is called cell-mediated. Another name for your humoral system is a dissolved system, your dissolved acquired immunity. Think of dissolved as just that. You've got chemicals, molecules that are floating around, dissolved, in the extracellular fluid. Extracellular being the key word here. This is outside the cell. This is an antibody-driven system. So you have antibodies floating around between the cells in your bloodstream. They're floating around in the plasma, for example. They encounter a pathogen that is also present between your blood cells. That pathogen has antigens on its surface with epitopes that attract specific antibodies. The antibodies are then going to grab onto those epitopes and act as, well, they're going to do a variety of different things, but one of the easiest ones to picture is the antibody latches on to the epitope on that antigen and acts as sort of like a neon sign, flashing, saying, invader, invader, invader. And that's going to attract phagocytic leukocytes, which will then come and engulf that pathogen. And it's just one example. This system is very, very effective if you have invaders in your extracellular fluid. Again, extracellular, outside of your actual body cells. Now you're going to need a lot of antibodies in order for this system to work effectively. And especially if you have pathogens introduced, say, into your bloodstream, if individual antibodies in your bloodstream have to wait before they encounter one of these pathogens, you may not have a high enough concentration of antibodies that respond to the antigens on that pathogen for one of them to encounter the pathogen quickly enough to mount a quick immune response. One solution your body has to that problem is the humoral system is mediated by these cells called B cells. Mediated means these are the primary cellular participants. They are going to drive a lot of these immune responses. B cells are antibody producers, mass producers. When B cells encounter a specific antigen, they start churning out huge amounts of antibodies that are targeted towards that particular antigen, flooding the bloodstream with those antibodies and dramatically increasing the odds that they will latch onto any other pathogens of that type that have made it into your body. Now that's in contrast to cell-mediated acquired immunity. This is an intracellular system. It defends against intracellular invaders. So if you have pathogens that are in your bloodstream but outside your body cells, they're pretty easy to find. But what happens when your own body's cells become infected and that infection is hiding inside of your own cells? This is where cell-mediated immunity comes into play. It's driven by these special cells called T cells. They're a type of lymphocyte. They're oftentimes called cytotoxic T cells. Think about that name. We know cyto means cell. Toxic generally means bad for you, dangerous. Cytotoxic T cells are cells that can destroy other cells. Okay? They can cause them to spontaneously die, what we call apoptosis. Apoptosis is spontaneous cell death. And T cells can cause other cells, like bacteria cells, or I'm sorry, like infected body cells, to spontaneously die or explode. T cells are really good at identifying any of your own body's cells that are infected. That's one of their primary jobs. And we'll talk about that more in a later slide. First, we've done a lot on this slide. There's a lot of blue writing here. Let's go back up to one of our first topics that I said we'd revisit, antibodies, up here. Let's talk about all the different functions that antibodies have. All right, there are 5 major functions of antibodies. I know this bullet list over here has 6 bullets. 2 of them are linked. Okay. Let's start at the beginning. One of the cool things that antibodies can do is they can neutralize a pathogen by making it unable to attach to host cells, essentially by dogpiling it. Okay. Neutralization, the example is down here. Antibodies attach themselves to any available epitope location on the surface of the pathogen. They essentially coat it until it's absolutely covered and it can no longer reach your body's cells to attach to them. It is too densely covered with antibodies. That's a pretty effective way to neutralize that pathogen, make it so it can't infect you. Next up is a similar idea, agglutination and precipitation. Precipitation is when you turn something into a solid or a semi-solid and cause it to sort of fall out or pass out of a liquid medium. Examples right here. Agglutination in this case is exactly what we saw when we were talking about blood testing. The antibodies latch onto the pathogens and cause them to clump together, making them much easier to filter out of your body. Okay. Next up, activation of complement proteins. Okay. That's up here. Your complement system, we're not going to talk very much about, but essentially your complement system is made up of a group of molecules, most of them are proteins, that can be activated to help facilitate and enhance your immune response. So these are frequently proteins that enhance your immune system's function. Oftentimes they cause inflammation. They can also help to promote cell lysis. Cell lysis is when a hole is popped, or a hole is created in the membrane of a cell, and the cell deflates like a balloon. Okay, it essentially gets popped very slowly, okay, leading to cell death. So, activation of complement proteins, this is linked to stimulation of inflammation on this list. All right, so these two guys go together. All right, next up. Antibodies can help attract phagocytes. That is right here. Gonna have to look close at these two boxes because they are rather similar. Okay. Attracting phagocytes is exactly what it sounds like. The antibodies act as that neon sign, attracting in things like macrophages to come and engulf the pathogen that the antibody has attached itself to. And then finally, antibodies can actually enhance phagocytosis. That's what you see here. It actually causes phagocytosis, the engulfing of that pathogen, to happen more efficiently and more quickly. All right. So those are the main functions of antibodies. Now let's talk about the cells that interact with them. The stars of the cellular show when it comes to adaptive immunity are your lymphocytes. Lymphocytes develop from what are called lymphoid stem cells, and they turn into one of three different kinds of immunity cells: natural killer or NK cells, B cells, and T cells. Now all of this starts in the red bone marrow. Okay? In your red bone marrow, there is a stem cell called a hemocytoblast. Hemocytoblasts are able to differentiate into these lymphoid stem cells. Now, not all lymphoid stem cells stay in the red bone marrow to develop into these leukocytes, leukocytes or lymphocytes. Instead, some of these lymphoid stem cells actually migrate to your thymus, which is an organ just deep to your sternum, right over your heart. Okay. Once in the thymus, this lymphoid stem cell is exposed to thymic hormones. And remember, hormones can trigger changes in cells. These hormones actually cause the production and differentiation of T cells. So it's exposure to hormones from the thymus that cause the lymphoid stem cell to turn into specifically our T cells. Okay. These T cells are then distributed into the bloodstream. Now, some of these lymphoid stem cells are going to be retained in the red bone marrow and they're going to mature there. They can mature into one of two types, either our B cells or our natural killer cells. Both of these cell types are also then going to be released into the bloodstream so that they can move to what we refer to as the peripheral tissues, meaning the rest of the tissues through the body. T cells, okay, function in cell-mediated immunity, meaning T cells are the ones that inspect your body's own cells to determine if they're healthy, and if they're not, they kill them. B cells participate in antibody-mediated immunity. Remember, B cells are the ones that are going to produce loads and loads and loads of specific antibodies when they're needed. And then finally, you've got natural killer cells, which is probably the coolest name for a cell in the body. You can call them NK cells, but why would you? Natural killer cells, they perform immunological surveillance. It's a really cool term. Immunological surveillance, it's what it sounds like. Your natural killer cells. So immunological surveillance is when natural killer cells patrol your bloodstream and looking for anything that registers as non-self. So they're another general hunter-killer cell that isn't looking for particular pathogens. It's looking for things that are presenting antigens that don't belong. What's kind of cool is natural killer cells do not need antibodies in order to locate cells that are infected. They can detect stressed cells without the use of those antibody signals or signs. They do this a couple of different ways. One of them is they can detect if a cell is presenting antigens on its surface. When one of your own body's cells is infected with a pathogen, sometimes it will grab antigens from that pathogen, and we call it presenting. It'll present them on the membrane, on its own cell membrane. Sort of like saying, I'm in trouble, I'm sick, here's a sample of what's in me. NK cells can detect those antigens directly, allowing them to identify the cell that's presenting them is sick and needs to be destroyed. The other giveaway that NK cells look for is that healthy cells produce a protein called MHC1. It stands for major histamine compatibility. Okay. Just know it's a protein. Healthy cells produce it. Unhealthy cells, like this one, don't. So if an NK cell, a natural killer cell, encounters a body cell that doesn't have this MHC1 protein on its surface, that's an indicator that there is something wrong with that cell. B cells, remember, these are your antibody producers. They are going to bind to an antigen, okay, and then, based on the characteristics of that antigen, they're going to start creating massive numbers of antibodies. First, the B cell changes into something called a plasmacyte. The plasmacyte is the version of the B cell that is an antibody manufacturing center, and it turns out huge numbers of antibodies that are specific to the antigen the B cell encountered. And these antibodies then attract our macrophages, and the macrophages, looking for that particular antigen based on the antibodies it encountered, is going to destroy any cells that have those antigens present on them. Now T cells, cytotoxic T cells, this is our last group. Remember, these guys mature in the thymus. That's one of the things that makes them unique. They respond to very specific antigens. And they generally have a memory for antigens that they have encountered before. So both T cells and B cells can develop memory for antigens that have been in the body before and that they fought off. Any antigen that the body recognizes, it likely already has antibodies for, and it will be able to mount a more immediate and a more rapid response the next time the body encounters that pathogen or that antigen. All right. The rest of this lecture we are going to dedicate to talking about the lymphatic system. And the lymphatic system does participate with immunity, and I'm going to show you where the overlap is. First off, what do we mean when we say lymphatic system? Well, the lymphatic system has 4 major functions. Before we get to that, though, I want you to look at the diagram. There's a lot of structural similarity between the lymphatic system and the system of blood vessels that run through your body. Lymphatic vessels and blood vessels oftentimes co-occur. You tend to find them together, right? So know that circulation of blood and circulation of lymph fluid are going to be tightly connected to each other. All right, let's talk about functions first. First, lymphatic system helps to maintain your blood fluid level. Remember when we talked about the ends of capillaries, the arterial and the venal ends, we talked about fluid exchange, fluid moving out of the capillaries, fluid moving back in, and that generally there tends to be fluid lost to surrounding tissues. Well, fluid lost at those capillaries tends to be collected by lymphatic capillaries, and eventually it gets returned to the blood so that the blood isn't continuously dehydrated. The lymphatic system helps to collect that lost fluid and then return it to the bloodstream. The lymphatic system is also really good at absorbing fat and any fat-soluble vitamins. It also helps to maintain the quality of your blood by promoting the filtration of by promoting the filtration of fluids in the blood, filtering out pathogens, microbes, anything that doesn't belong, which helps purify the blood. It also promotes the purification of red blood cells so that old red blood cells get pulled out and replaced by new ones. Also, here's that immune system connection. The lymphatic system helps to protect you against infections. Lymphocytes and monocytes are both strongly affiliated with this system. All right, so let's talk about lymphatic flow and what's actually flowing through these vessels. Well, we give a special name to the fluid from the blood that leaks into the surrounding tissue at the capillary sites. We call this leaked fluid lymph. Okay? When lymph is collected by these lymphatic vessels, it moves in one direction through the vessels. It starts at the capillaries, so lymphatic capillaries collect all of the lymph that's being lost from your blood vessels' capillaries. Those capillaries then join together to form vessels. The vessels join together to form nodes. Those nodes then act as a filtration site. We'll talk about them later. They then transfer large volumes of lymph into ducts, which are sort of a combination of a gateway and a storage site for large volumes of lymph. The ducts then reintroduce the lymph back into the bloodstream at the subclavian veins. Subclavian meaning below your clavicle. After that, the lymph winds up back in the bloodstream. As I mentioned earlier, lymphatic vessels, they travel alongside blood vessels, and structurally they're pretty similar to veins. They have one-way valves, just like veins do. Oops. So they have those one-way valves to prevent backflow, and their unidirectional flow is facilitated, it's helped along by body movement and muscle contractions. So a lot of similarities with veins here. All right, so those lymphatic capillaries coalesce into lymphatic vessels, and those lymphatic vessels then join together to form what are called lymphatic trunks and ducts. The ducts are what I want you to pay particular attention to. There are 2 of them that you need to worry about. The first is called the right lymphatic duct, and it's right here, okay, just underneath your right subclavian vein. The bigger of the 2 is called the thoracic duct. It's right here underneath your left subclavian vein. Now remember, these ducts are going to empty straight into the subclavian veins, so the locations make sense. Now if you notice in the drawings, the thoracic duct is significantly larger than the lymphatic duct. And the reason is the thoracic duct is responsible for draining the entire lower body. In fact, there has to be another sort of a stopover point along the lymphatic trunk on its way from the lower body to the upper body, because there is so much lymph being accumulated and sent upwards. There sort of has to be a stopover point for it, a holding point. That holding location is down here. Okay. It's called the cisterna chyli. It's an enlarged sinus, so basically a really, really large area of these vessels that accumulates a lot of lymph until it can be sent upwards to the thoracic duct and then dumped into the subclavian veins. So just to give you a sense of relative flow here, the right lymphatic duct, which drains your right arm, the right portion of your head and your right chest, it's draining about 20% of the lymph in your body back into your subclavian veins. The thoracic duct, on the other hand, is draining the other 80%. It's responsible for the whole rest of the body. Okay, so it's significantly larger in terms of its responsibility. All right, what we're going to talk about now are some important physical features of the lymphatic system. Specifically, the ones that participate in immunity are lymph nodes. Lymph nodes we tend to associate with being maybe at the back of our neck, on the sides of our jaw, maybe even in our armpits. But lymph nodes are actually scattered throughout the body. Those just happen to be a few that are superficial enough that we can palpate them. The only place you won't find lymph nodes is in the brain. These lymph nodes are attached to your lymphatic vessels, and they actually act like a filter for lymph. Okay? So these lymphatic vessels are going to dump into your lymph nodes. Okay? And any abnormal cells present in the lymph, any pathogens, anything else that should not be there, is going to be filtered out by the lymph node itself. There are actually lymphocytes and monocytes that are permanently attached to the inside of the lymph node, and their job is to just inspect the lymph and make sure nothing is in it that shouldn't be there. Right? So this is where that cleaning of your blood supply comes in. The lymph that gets reintroduced into your bloodstream in your subclavian veins is going to be more pure than the lymph that was released from your blood at the capillary sites. All right. Next up, we're going to talk about a few organs that participate in the lymphatic system. The first one we've already mentioned, the thymus, is this big white structure here. Okay? It's located superior to the heart and deep to the sternum. Remember, this is where your immature lymphocytes go to become cytotoxic T cells in response to hormone exposure. Your thymus actually grows until puberty, and then it slowly starts to atrophy during your adulthood. Next lymphatic structures you need to know about are called tonsils. Tonsils are aggregations of lymphocytes that help to remove pathogens from either the air you're inhaling or the food you're consuming. For that reason, your tonsils are located along your airways and at the back of your pharynx. The ones at the back of, or the ones that participate most in filtering out pathogens from the air you're inhaling are called the pharyngeal tonsils. They are straight back from the nose. You can see them here in this diagram. Okay. Right there. So directly behind your sinuses, you're going to find these pharyngeal tonsils. The ones that are responsible for filtering out pathogens from food and also from air are your palatine and your lingual tonsils. The palatine tonsils are at the back of your, think of it as your palate. So we talk about having a palate that's sensitive to certain foods. Think palatine being at the back of your palate, the back of your mouth. And then lingual, it's at the base of your tongue. I think lingual is in tongues. When your tongue ends down at the back of your mouth, beginning of your throat, that's where your lingual tonsils are. Okay? And finally, the spleen. Your spleen is located in your upper left abdominal cavity. It can store things like platelets and blood cells, but the function of the spleen that we are most concerned with is the fact that it filters your blood. Right? So if you look at this cross section of the thorax here, this is a little bit weird. It's a perspective we don't normally have. We have cut the body in half horizontally, and we're looking down into the lower abdomen. So you see the liver here. This is your stomach. Okay. Here's the spine, right? And on either side of that, you've got your kidneys. The aorta is here, so it's running up and down. We're just seeing a cross section of it. And then over here we have the spleen. All right, so it's this nice purple structure. Now the spleen filters lymph, but also blood, both fluids. It consists of two tissue types. We call them red pulp and white pulp. They have a number of different functions. What I want you to worry about is the fact that red pulp removes old or damaged red blood cells. Remember, we define old for a red blood cell as being 120 days. White pulp, on the other hand, participates in the removal of pathogens from the blood. We're just going to leave it at that. All right, that's it for immunity and the lymphatic system.